NEET aspirants, this is your Prasanna Ma'am from NEET World, and here we are with our 100 topmost important concepts for NEET 2026. And today we are going to discuss about Bryophytes and Pteridophytes under plant kingdom, which are utmost important concepts where we can expect at least 2 questions in our final NEET examination. So yes, first let us look into Bryophytes. In this, if you have to look into, Bryophytes are said to be the most primitive land plants, and they are said to be amphibians of plant kingdom because though they are in shade and dampy places, they rely on water for sexual reproduction. And then Bryophytes, they are archegoniate. First archegonia is observed in Bryophytes itself, and they are embryophytic, atracheophytic cryptogams. Atracheophytic, which means the absence of tracheids and vessels. Okay, so they are atracheophytic cryptogams, no vascular bundles are present. You can also call it as non-vascular terrestrial cryptogams. All these synonyms are important for Bryophytes, right ma? Now if you have to look into its habitat and habitat is most shaded areas like hills, damp, humid, shaded localities, these Bryophytes are present. And there are no true root, leaf and stem like structures, but then we can identify root like rhizoids. Rhizoids can be unicellular rhizoids or multicellular rhizoids. Unicellular rhizoids, you can see it in liverworts, multicellular rhizoids in mosses, fine, and leaf like phylloids, stem like colloids are present. And then plant body is differentiated than algae, but that is not well differentiated into root, stem and leaf. The main plant body in Bryophyte is gametophytic plant body, which is haploid. This gametophyte will be having multicellular sex organs, both antheridium and archegonium. Okay, antheridium and archegonium, which are multicellular sex organs. This antheridium will be having jacketed layers. Inside antheridium, androcytes are present. These androcytes are also called as spore mother cells. Okay, androcytes are also called as spore mother cells, and it is having a stalk. This spore mother cells or androcytes will be releasing out anthrozoids. This anthrozoids through zooidogamous oogamy, it will reach into the egg cell which is present inside the archegonium. Archegonium is also a jacketed sex organ where it has neck and neck canal cells, venter canal cells. Below the venter canal cell we have egg, which is also called as oosphere. The flagellated anthrozoids will reach this egg or oosphere through zooidogamous oogamy. Clear, right ma? Next, where is the site of fertilization happening? Site of fertilization will happen inside the archegonia, that is with oosphere or egg cell. This happens inside the archegonium. Site of the fertilization is archegonium, which is very important, do remember. And the product of fertilization is zygote. Zygote is the product of fertilization. This is differentiated into embryo. Embryo matures into a diploid sporophyte. Clear, right ma? So here antheridium is haploid or antherozoid is haploid. Oosphere or egg is haploid. Then it results in the formation of diploid zygote, which is differentiating into diploid embryo. Later this diploid embryo will mature into the diploid sporophyte. Till here it is clear, right ma? Up to gametophyte. Now we will look into the sporophytic development. Sporophyte, it is attached to the gametophyte for nourishment. Okay, so it is parasitically dependent on gametophyte for nutrition, wherein spore mother cells from the sporophyte, which are diploid, undergo sporic meiosis. This will undergo sporic meiosis. Whereas in algae, where we have seen the primitive plants, that is algae, zygotic meiosis takes place, but here in Bryophytes, sporic meiosis occurs, which helps in the release of haploid spores. And all these spores are homosporous only. There is no differentiation into microspores and megaspores in Bryophytes. Only single type of spores are present. Those are homospores. This homospores landing onto the suitable substrata First they will develop into a thalloid gametophyte if it is in liverworts, or to a protonema which is a filamentous state in moss. Okay. So, sporophyte is dependent on the gametophyte for nutrition, where inside sporophyte, sporophyte is of course diploid, right? Inside sporophyte, spore mother cells will be there which are diploid, which undergoes sporic meiosis, and that helps in the release of haploid spores. Haploid spores are homosporous in condition. This will develop or differentiate into thalloid gametophyte in liverworts and protonema, which is a filamentous stage in mosses. Then Bryophyta classification. Bryophyta division is classified into 3 classes. Those are Hepaticopsida. Best example that we are learning under Hepaticopsida is liverworts, and then Anthoceratopsida, hornworts, Bryopsida, mosses. Okay. So, these 3 are the 3 classes of Bryophyta. First, let us clearly look into the liverworts. Habitat of liverworts are again moist and shady, mostly in the bank of streams, marshy grounded, damp soils, bark of trees and deep inside the wood. This is the habitat of liverworts. Habit of liverworts include the plant body is thalloid plant body. Best example of liverwort is Marchantia. Okay. So, let us clearly learn how the thallus of liverwort is. Thallus of liverwort is dorsiventral like this, which is closely appressed to the substratum, and this shows dichotomous branching clearly. Okay. Dichotomous branching is observed, and this dorsiventral thallus is closely appressed to the substratum, and this is dorsiventral. Clear, right? This is the thallus description of Marchantia. If you have to see one more liverwort, that is Sporella, the leafy members will have tiny leaf-like appendages like this. Okay. Tiny leaf-like appendages are present, which are in 2 rows in the stem-like structures. Okay. Then vegetative reproduction. Vegetative reproduction is through fragmentation and gyma. Okay. So, these are gyma which are present inside the gyma cups. Here if you can see gyma clearly, gyma which is present in gyma cups. What is the description of gyma? These are green multicellular asexual buds, green multicellular asexual buds which are present inside the gyma cups. They aid in vegetative reproduction. They develop on the small receptacles called as gyma cups. They mature gyma will detach from the parent body and they will germinate into a new Marchantia individual. Clear, right? They will detach and they will fall onto the suitable substrata, develop into a new individual. Next, sexual reproduction. Sexual reproduction, we have to learn first what is homothallic and what is heterothallic, and then we can look into the sexual reproduction types. What is homothallic or monoecious? If both male and female sex organs, that is antheridia and archegonia, are present on the same plant body, then it is homothallic or monoecious. But then if they are present on 2 different plant bodies, like how you can see in Marchantia, antheridia is present on one thallus, archegonium is present on the other thallus. This is called as dioecious or heterothallic. Most important segment to be noted where we can expect a questionnaire from here. So, this is about Marchantia. Marchantia, this antheridiophore, the stalk which is carrying antheridia is called as antheridiophore. They will release out antherozoids through elaters. They will be having elaters over here. With the help of elaters, they will be releasing out antherozoids, and archegoniophore will bear egg, and here again the site of fertilization is archegonia, and the product of the fertilization is zygote, which is differentiated into embryo that matures to a diploid sporophyte. This matures into a diploid sporophyte. This is all about the sexual reproduction. Now, hornworts. Thalloid consists of rhizoids, elongated horn-like appendages, and sporophyte here in hornworts contain capsule, intercalary meristematic zone like this. Okay, intercalary meristematic zone and a foot. So, it has foot, intercalary meristematic zone and capsule. Capsule will be having spores. To disperse the spores, elaters are absent, but then they have pseudo-elaters. Okay, pseudo-elaters are present in hornworts. Best examples of hornworts is Anthoceras. Okay, this comes under the class Anthoceratopsida. Now mosses, okay, mosses of bryophytes. Gametophyte will be exhibiting two stages. One is juvenile stage and the second is adult leafy stage, which bears multicellular sex organs, Antheridium and Archegonium. So the juvenile stage, which is protonema, is formed whenever the haploid spores which are released from the capsule, okay, haploid spores which are released from the capsule falling onto the suitable substrata, first they will develop into a creeping, green, branched, frequently filamentous stage. This creeping branched filamentous stage is called as protonema, which is the juvenile stage. This directly forms out what? Spores, and that will develop into a leafy stage, which is upright slender axis, and this bears spirally arranged leaves, okay. And the apex will be having Antheridia and Archegonia. Apex will be having what, ma? Antheridia and Archegonia. Fine, till here it is clear, right? Next, they will bear multicellular uniseriate filamentous sterile hairs called as paraphyses. So this leafy gametophyte stage will be having what? Multicellular uniseriate filamentous sterile hairs. These are called as paraphyses. And sporophyte will be bearing capsule, seta and food. Sporophyte will be having food, seta and capsule. Funaria is the best example over here. This is the image of what? Funaria. So here leafy gametophyte stage is there. From this leafy gametophyte stage, sporophyte will be evolved. This sporophyte, foot of the sporophyte is attached to the leafy gametophytic stage. So here if you have to mention the ploidy from this diagram, sometimes this diagram will be given and they will be asking us for the ploidy. We know the gametophyte stage over here is haploid and sporophyte stage is diploid. What are the gametophytic structures over here, ma? Rhizoids are the gametophytic structures which are haploid. Main axis is haploid, leaves are haploid, whereas foot, seta, capsule, these are the structures of sporophyte. They will be diploid. Diploid seta and diploid capsule. Inside this capsule, spore mother cells will be present. Spore mother cells will be undergoing what meiosis? Sporic meiosis. Okay, this will be undergoing sporic meiosis inside the capsule, and haploid spores will be released out. This haploid spores are released out through what? Elaters. Clear, right, ma? So more elaborate than in liverworts, sorry. This spores, haploid spores will be released by peristomic teeth. In liverworts though, the spore dispersal is by elaters. In mosses, for example Funaria, the spore dispersal is mediated by peristomic teeth. Remember that, ma. So here spore mother cell undergoes sporic meiosis. Haploid spores will be released through peristomic teeth. And clear with the ploidy also, right, ma? Next. So here if you see, the best examples of mosses in bryophytes include Funaria, which is commonly called as cod moss, Sphagnum, which is called as peat moss, Polytrichum is called as hair cap moss. Clear? Now let us look into the economic importance of bryophytes. Mosses play an important role, especially in the plant succession. Of course, we know that lichens are the pioneers of plant succession. After lichens, the complete bed is formed by what? Mosses. Mosses, after lichens, they mediate the process of plant succession. They play a crucial role in plant succession after lichens, and they will prevent soil erosion. Sphagnum of bryophytes have highest economic use. Many at times the uses or the economic importance of sphagnum is asked in our NEET exam beta. So sphagnum is also called as cotton moss, bog moss and peat moss. Cotton moss is it is called as cotton moss because the dry sphagnum is used to transport the seedlings and cut plants. Okay, this dry sphagnum is used to transport the seedlings and cut moss. Bog moss is used to build the soil in rock crevices, and this will fill ponds and lakes. Peat moss is used as good manure and fuel. Clear, right? So this is about the economic importance of bryophytes. Let us once quickly revise the complete life cycle of bryophytes. Okay. So here if you see let us start with the dominant plant body or the main plant body, that is haploid gametophyte. This is having both stages, right, juvenile stage and adult stage. The juvenile stage is what? Haploid protonema, which is green, branched and filamentous. So this protonema undergoes budding and that will lead to the formation of an adult leafy gametophyte, which bears the multicellular sex organs Antheridia and Archegonia, which are jacketed, and Antheridia or antherozoids will be released through pseudogamous oogamy. They will reach the archegonium and the fertilized egg or oosphere. After fertilization, diploid zygote is formed, which will differentiate into diploid embryo. This diploid embryo will mature into diploid sporophyte. In the sporophyte, at the capsule region, spore mother cells are present which are diploid. They will undergo sporic meiosis. Okay, sporic meiosis, and after this sporic meiosis, haploid spores will be released and this falling onto the substrata develop into protonema. So here you can see equal stages of haploid and diploid stages. So the life cycle of bryophytes will be haplodiplontic life cycle. Clear, right ma? Next. Now let us start the clear and brief understanding of pteridophytes. First let us look into the synonyms of that. These are the first terrestrial plants to possess vascular tissues. Okay, pteridophytes are the first terrestrial plants to possess vascular tissue. They are embryophytic, archegoniate, tracheophytic, seedless vascular plants. Okay, first embryo are seen, first embryo formation is observed in, of course, bryophytes, but after that it is pteridophytes. In this bryophyte will be sustaining in the female gametophyte structure, which show precursor to seed habit, and then a archegoniate, tracheophytes, these are the first tracheophytes, seedless vascular cryptogams. Okay, vascular cryptogams are only pteridophytes, non-vascular cryptogams are bryophytes. If you look into the habitat of pteridophytes, they exist in cool, damp, shady places and sandy soil conditions. Here in pteridophytes, the main plant body or the dominant phase is sporophyte. In bryophytes, dominant plant body is gametophyte, but here it is sporophyte. And sporophyte is independent of gametophyte here. In pteridophytes, sporophyte is independent of gametophyte. And gametophyte is also independent of sporophyte. Both are independent to each other in pteridophytes. Dominant plant body is sporophyte. This includes club mosses, horsetails and ferns. Mosses in bryophytes, ferns in pteridophytes. So as we have discussed, main plant body is sporophyte, which is clearly differentiated into true root, stem and leaves. Roots are mostly adventitious types and leaves are of two types, microphyllous leaves and macrophyllous leaves. Microphyllous leaves are small, for example Selaginella. Macrophyllous leaves are large, example ferns. Okay, microphyllous leaves are small, for example Selaginella. Macrophyllous leaves are large, for example ferns. Here there is one more point to say, that is leaf structures will be bearing spores in the pteridophytes. As such, the leaves over here are called as sporophylls. Leaves are called as sporophylls. Clear, right ma? Next. Stem, internal anatomy will show epidermis, cortex and stele. What type of stele is observed in pteridophytes? Primitive form of stele, that is protostele, where in protostele you can observe at the central region vascular bundles are present, that is xylem and phloem, which is covered by the cortex and epidermis. Clear, right? So this type of stele, primitive stele, is called as what? Protostele. Yeah, as I said you, leaves and leaflets will bear what? Sporangia, that is why they will be calling as sporophylls. Based on the development of sporangia, we have two types of sporangiates. One is leptosporangiate if the development of sporangium is from one single cell. If it is from one single cell, you will call it as leptosporangiate. If it is from group of cells, then it is called as eusporangiate type of development. Sporangia which is diploid or spore mother cell, after sporic meiosis inside the sporophyte, they will be releasing haploid spores. Unlike in bryophytes where we have seen only homosporous condition, in pteridophytes we will be seeing heterosporous condition. So, the first heterosporous condition is observed where ma? In pteridophytes only. In pteridophytes, first heterosporous condition is observed. So, both the type of spores, homospores and heterosporous condition is observed. Heterosporous, you will see two types of spores, microspores and megaspores. Microspores will be developing into a male gametophyte, megaspores will be developing into a female gametophyte. Best examples to show heterosporous condition are heterosporous condition, Marsilea, Azolla, Selaginella and Salvinia. This shows heterosporous condition. Remember that is very important. Examples many at times asked with respect to heterosporous condition in pteridophytes. Marsilea, Azolla, Selaginella and Salvinia. Of course, Selaginella and Salvinia are anyhow highlighted in your NCERT. Right ma? Next. So, homosporous condition where the gametophyte which is developed from homosporous type of spores are monoecious, or homothallic where both the male and female reproductive structures will be on the same plant body, whereas heterosporous species will be dioecious as we have discussed. Microspores will develop into male gametophyte, megaspores will be developing into a female gametophyte. And then strobili or cones, sporophylls form distinct compact structures over here. Okay, sporophylls, the spores which are bearing, the leaves which are bearing spores, those are sporophylls, right. If they are turning into compact structures, these are called as strobili or cones. The strobili or cones are observed in Selaginella and Equisetum. Okay, strobili or cones are present in Selaginella and Equisetum. Clear, right? So, those stalks which are bearing this strobili are called as sporangiophores. Okay. Then the spores which are haploid landing onto the suitable substratum, they will be developing into what? First, juvenile stage in pteridophytes, those are prothallus. Prothallus, how you can describe it? As small, inconspicuous, multicellular, free-living, photosynthetic thalloid gametophyte. Okay. Prothallus is well described as small, inconspicuous, multicellular, free-living, photosynthetic thalloid gametophyte. You always have to remember the description of Protonema and Prothallus. Many at times the questions are asked on this too. Protonema, which is the primary gametophytic structure in bryophytes, Prothallus, which is the primary gametophytic structure in pteridophytes. Clear, right ma? Next, this gametophyte will be bearing sex organs which are multicellular, jacketed and sessile. So, this heterosporous condition, megaspores and microspores. Megaspores, they develop into female gametophyte, microspores into male gametophyte. So, now if you have to see once the life cycle, it is of course the sperm will fertilize with egg or oosphere by what? Zooidogamous oogamy, and here a zygote after developing into embryo, they will sustain for some time in archegonia. This habit is called as precursor to seed habit. Seed is not formed, but the habit of seed formation is shown. Okay. So, this is the precursor to seed habit, which is an important step to evolution, connecting what? Cryptogams to phanerogams or spermatophytes. So, if you have to describe sporophyte over here, that is multicellular, well differentiated and dominant phase in pteridophytes. And here pteridophytes will be having 4 classes: Coelopsida, Example, Coleotum, Lycopsida, Selaginella and Lycopodium, Spinopsida, Equisetum, Pteropsida, Bryopteris, Pteris and Adiantum, which is called as walking fern. So, all these 4 classes and examples are important. We can expect a match the following type of question from this area. Okay? And next about Bryopteris. This is useful for MCET. Okay? Description about Bryopteris, and this description about Bryopteris is useful for MCET students. You can listen to this. Where the stem of Bryopteris is underground rhizome type of stem, leaves are large, aerial and they are pinnately compound, which are called as fronds. Petioles, if you have to see, petioles, they are covered by multicellular hair-like structures called as ramenta. Leaflets are open dichotomous leaflets which show furcate venation. Okay, they will show furcate venation. Sporangia are formed on the ventral side, not on the dorsal side, they are formed on the ventral side, and you call this as what? Sori, as they are group of sporangia, so you call it as sori. Antherozoids are spirally coiled and multiciliate. Okay, antherozoids are what? Spirally coiled and multiciliate. Each sorus is covered by membranous sheath with true indusium. Each sorus is covered by a membranous sheath which is true indusium. Pteris, if you have to see, in pteris, each sorus is protected by a reflexed margin like this. So, each sorus is protected by a reflexed margin of fertile leaflet. So, this is called as what? False indusium. True indusium is observed in Bryopteris. False indusium is observed in pteris. Just remember that. Now, lastly, the economic importance of pteridophytes. Economic importance of pteridophytes, they are used as what? Soil binders. They are used in medicinal purposes. For example, Bryopteris is used as the phytochemicals of Bryopteris are used as what? Anti-helminthic drugs. Equisetum stems are used in scoring, polishing of metals. So, this is all about Bryophytes and Pteridophytes. In our next class, we will be focusing on Gymnosperms and Angiosperms from this chapter. Okay? Thank you, and that is all for today.